Hi guys, let's do 6.3, metabolic pathways and enzymes. Reactions, chemical reactions normally occur in a sequence where the products of an earlier reactor earlier reaction become the reactants, also known as substrates of a later reaction. Such linked reactions form a metabolic pathway. It begins with a particular reactant and then it goes through several intermediaries and then you get to a particular end product. So in other words, the product of the first reaction is the substrate of the second, and so on and so on and so on, except in the case of letter G, which is our end product. So A is what we start with. We go through one reaction, we get to B. We go through another reaction, we get to C. We go through another reaction, we get to D. Another reaction, we get to E. Another reaction to F, and then from F to our final product. So that is an example of a metabolic pathway. Enzymes function as a catalyst. They are made out of proteins, and the reactants of an enzymatically catalyzed reaction are called substrates. Each enzyme accelerates one specific reaction. It has one job, and it will go on doing that one job until it gets a signal telling it to stop. Each reaction in a metabolic pathway requires a unique and specific enzyme. The end product will not be formed unless every single enzyme in the pathway is present and functioning the way that it should. So this reaction down here from A to G, we will not get to G if even one of these enzymes is not functioning properly. It's like knocking over dominoes. If we were to take out E3, E4, E5, and E6 are not going to be able to do their job. Those dominoes are not going to fall over. Enzyme substrate complex. The active site of an enzyme complexes with the substrates. It causes that active site to change shape, and the shape change forces substrates together, initiating bond formation. So basically, the reactants come to the enzyme, okay? The active site changes shape, and the two things come together and form our final product. Or in some cases, the substrate for the next one. Induced fit model, enzyme is induced to undergo a slight alteration to achieve optimum fit for the substrate. So the enzyme changes its shape just a little bit so that it's easier for the substrate to fit. Degradation. Enzyme complexes or bonds with a single substrate molecule, and then it is broken down into two different molecules, which are then released. So that is a degradation reaction. Synthesis is when the enzyme joins or complexes with two different substrates and joins them together and releases a single product. A single molecule is made. So here we have a diagram. Here we have a degradation. And here we have a synthesis. All right, so we start with a substrate. It bonds with the enzyme. The enzyme breaks it apart into two different molecules, and the enzyme goes on and does it again. Down here we have a synthesis. Two substrates go into the enzyme. They are bonded together, then they are released from the enzyme, and the enzyme goes on to do it again. Energy of activation. Molecules frequently do not react with one another unless they are activated in some way. The energy must be added to at least one reactant to initiate the reaction. That's called the energy of activation, or EA. Prevents molecules from spontaneously degrading in the cell. So they have to have a certain energy input for that reaction to occur. Enzymes operate by lowering that energy of activation. This is accomplished by bringing substrates into contact with one another because they might not bump into each other randomly throughout the cell. So the enzyme brings them together so that those reactions can occur much more easily. This influences the rate of reaction and is why enzymes are catalysts of chemical reactions. So here we have an energy activation chart or graph. So the orange line is when the enzyme is present, and you'll notice that the amount of energy required is a lot less than when the enzyme is not present. Factors affecting enzymatic speed, the substrate concentration. Enzyme activity increases with substrate concentration due to more frequent collisions between substrate molecules and the enzyme. That makes sense because if there's more of it there, you're more likely to bump into it. Temperature. Enzyme activity increases with temperature. Warmer temperatures cause more effective collisions between the enzyme and the substrate. However, hot temperatures, if it's too hot, they can denature and destroy that enzyme. Siamese cats have a mutation that causes enzymes to be active at cooler temperatures that affects their coloration. pH can also influence the rate that enzymes act. In the case of pepsin or trypsin, optimum protein digestion takes place at the respective pH optima. So they have a range of pH where they work the best. So here we have a graph showing the rate of reaction as a function of temperature. So there's an optimum temperature about 40 degrees for this enzyme to work. Okay, body temperature of ectothermic animals often limit reaction rates. So cold-blooded animals, whenever it gets cold, their body can't go through the reactions, and normally they will hibernate or do some other kind of process similar to hibernation. The effective pH on rate of reaction. So here we have pepsin, and you'll notice that its optimal pH is very, very low. Okay, it is about 2. And here we have trypsin, and its optimal pH is about 8. So those enzymes act at different pHs. Factors affecting enzymatic speed. Cells can regulate the presence or absence of an enzyme. They can regulate the concentration, and they can also activate or deactivate those enzymes. Some enzymes have cofactors. Those are molecules required to activate the enzyme. FAD and FAD+ and NADP+ are cofactors. The first two in cellular respiration and the third in photosynthesis. So these are enzymes that are needed to trigger the enzyme to do its job. Coenzymes are non-protein organic molecules. Vitamins are small organic compounds required in the diet for the synthesis of coenzymes. So it is important to get all of your vitamins so that your enzymes can act the correct way. So here we have a picture of a Siamese cat. Isn't she pretty? Her coloration is affected by the enzymes that are present, and they work at a cooler temperature than other cats. Cofactors at the active site. So here we have our enzyme. Here we have the cofactor at the active site. The substrate will bind with that active site. All right, see how it bonded right there. Enzyme inhibition, reverse enzyme inhibition. A substance known as an inhibitor binds to the enzyme and decreases its activity. There are different kinds of inhibition. There's competitive. The substrate and the inhibitor are both able to bind to the active site, and they compete with one another. The product forms only when the substrate binds to that active site. In non-competitive inhibition, the inhibitor does not bind to the active site, but an allosteric site, so somewhere else on that enzyme. A change in shape initiated by the inhibitor binding to the allosteric site changes the shape of the active site, making it unable to bind to the substrate. So non-competitive bonds with the enzyme somewhere else other than the active site and changes its shape. So here we have a diagram of non-competitive. So we have A going in to our enzyme and making our product. So just take a look at that. It's in your notes too, guys. Enzyme inhibitors can spell death. So there are certain substances that change the shape of enzymes that change the way that your body works. So materials that irreversibly inhibit an enzyme are known as poisons. Okay, cyanide is a, I don't want to say popular one, but one that a lot of people know about. And it inhibits the enzyme required for ATP, which is energy production. Sarin inhibits an enzyme located at a neuromuscular junction, which means your muscles don't work right. And warfarin inhibits an enzyme responsible for blood clotting processes. So warfarin is used as a blood thinner. My grandma was on it before she passed away, and she would just wake up with bruises. She would scratch herself in the middle of the night, and she would wake up with massive bruises because she couldn't clot properly. So it was particularly dangerous if she fell because she could develop very serious internal bleeding that would be very difficult to stop. And that is it for these notes, guys. If you have any questions, let me know.